Okay, so dihybrid inheritance. What is the difference between monogenic inheritance and dihybrid inheritance? So to understand the dihybrid cross, it's necessary to understand the processes of meiosis. So in the first meiotic division, the homologous pairs, which are here, or actually here, the homologous pairs of chromosomes are aligned at random before separating from each other. The separation of the chromosomes gives rise to the independent assortment of the chromosomes. This happens, independent assortment happens, we have to go over that in a different video. In the second meiotic division, the 4 cells that are developed to develop into haploid gametes. So all genetic crosses show how characteristics are inherited through the generations. So what is the difference between a monogenic inheritance and a dihybrid inheritance? So a monohybrid cross shows the inheritance of one characteristic through 2 generations, so from paternal and maternal to offspring. So you see the one characteristic go to a different, to the offspring. So that's 2 generations of one characteristic. So parents to the F1 generation, and then from the F1 generation to the F2 generation. A dihybrid cross, on the other hand, shows the inheritance of two characteristics through 2 generations. So in a dihybrid cross, the genes that determine the 2 characteristics are located on the different pairs of homologous chromosomes. Each of these genes can have 2 or more alleles. So for example, these alleles can occur, so this is one pair of alleles that are on a set of chromosomes, but they can also have another one, which is on the pair of chromosomes. There's 2 alleles that code for different characteristics on the same chromosomes. So, an example of this is peas or seeds. So if I do, get my green pen out. So you can have green peas, or seeds, green seeds, which is Y. You can have yellow seeds, that's a little Y, it should be a little Y, that should be recessive. So the green seeds are recessive. I'm going to do the yellow and orange. The yellow seeds are dominant. The round seeds, so that you've got colour and you've got a shape. So the round seeds, that's not very round, but the round is dominant, so that's a big R, and the wrinkled is recessive. So wrinkled is little r with recessive. So, for example, we now have one yellow round, I'm putting yellow as orange, so we have one yellow parental is one yellow round, so that would be dominant Y and one dominant R, yellow round, and a green wrinkled. So that's the green wrinkled. So that's 2 recessive alleles as the gametes. So these that's the parental phenotype, and then the parental genotype would be, say if this is homozygous, you would have 2 dominant alleles for yellow, and then 2 dominant alleles for round, and then you'd have 2 recessive greens, and then 2 recessive wrinkled. So you've got that. They'd have to tell you in the question that you'd have homozygous. Unless it's recessive, then you'd obviously have to have homozygous. So this is the parent's phenotype, and then you have the parent's genotype here. So then it goes through the process of meiosis, which you put the process of meiosis here. It's through meiosis. And this is a true breeding of homozygous pairs, which we've explained before. It's a true breeding because the offspring is either going to have either express one of these characteristics, because it's of the one of the characteristics of the parent, because it's the same. It's got the same homozygous pairing, so it's either going to be green wrinkled or yellow round. It can't, because there's no carriers. There's nothing hiding, basically. So, and then we have the gametes. These gametes are, I'll do them in brown. We have big Y, capital Y for dominant yellow, no don't, it's not, sorry, ignore that. Dominant Y for yellow, a dominant R, and then we have a dominant Y, dominant R. So these are the ones that you can get off this one. I've kind of done it badly. But between the two dominants and then the two dominants, you can only make dominant Y, dominant R, dominant Y, dominant R. That's all you can get from that one. And remember, when you do gametes, you have to circle them. So you circle the gametes. And then from this one, what can we get? We can get recessive Y, recessive R, and recessive Y and recessive R. That's all you can get. There's no dominance, so you just have to repeat what you've got there. YR, and then another YR. So these are from these two here. So from these two, and then these two are from the yellow one. And that's the gamete, so always circle the gametes. And remember, this is the F1 generation, because this is the first generation that we're finding out about. All of the offspring in the F1 generation have heterozygous genotypes. So, as you can see, all of these here, heterozygous, they're all heterozygous. Yeah. Wait, no, you can find the heterozygous when you do your table. So you do Y, R, Y, R, then you do little y, little r, little y, little y, little r. So then you cross this over. I'll just do one because they're actually all the same. So to find out what the offspring would be, it would be capital Y, capital R, which is both dominant, and then it would both have the two recessives. So all of them, you would find out to do it properly, but all of them, 100%, would have capital Y, capital R, little y, little r, two dominants, then two recessive. This means it's heterozygous genotypes, which is, so overall, 100% is big Y, big R, little r. So because there's a dominant colour, the dominant colour here is yellow, so it 100% would be yellow, and then 100% would also be R, which is round. So 100% would be yellow and round. However, they would also be carriers for the next generation. So you wouldn't have another set of true breeding, because it would not be both. It wouldn't always express, because you would be able to get one of the recessive alleles to be expressed in the F2 genotypes. Right. So following it through. Let's put this on here. So as you follow it through, so this is the F2 genotypes. You have two yellow rounds, because that's what we've just produced, and then it goes through meiosis again. Then remember to circle the gametes. So whatever can be made from what the genotype was of the parents. So try all of the possible gametes that could be made. So you can have a capital Y, a little r, a little y, big R, etc., etc. So you can see there what different types of gametes you can make. So the gametes that come off the ones from before. Then you put that into a table, so you have the gametes lined up. You put the gametes in the pair, so the capital Y, capital R, etc., etc. So this is the offspring of the F2 genotypes, and the offspring, so we'll do one, say we want to do one here, this will be capital Y, capital R, which we do with one big R, one Y, so you have capital R, capital Y, little y, capital R, little r. So that would actually come out as how we've done it before, but the offspring is going to be 9 yellow seeded, 3 yellow wrinkled, 3 green round, and 1 green wrinkled. Right, this is a very important ratio. I'll just explain this ratio. So this ratio of 9 to 3 to 3 to 1. This is something called it's an expected ratio. So why is this an expected ratio? This is for two reasons. Number one, fertilisation of gametes is random. So fertilisation of gametes is random. And number two, the gene pairs assort independently during meiosis. So the gene pairs assort independently during meiosis. Right. So if you get a question about why is there the general ratio of, the expected ratio of 9 to 3 to 3 to 1, this is because the fertilisation of gametes is random, and number two, the gene pair assorts